just to recap for you where we left off, we were introduced into Chapter 10, which is setting us up for this concept of inheritance, and we identified this is probably the more interesting part for biology class. As a student who's not a biology major, we can actually see the genetics in practice here, looking at how traits are inherited from one generation to another. And at some point I'll be able to realize how fast this semester is going. The last time which we met, we talked about the basic traits, and we talked about some terminology. We talked about dominant versus recessive alleles. We differentiated, redefined the term gene, a chromosome, a homologous chromosome. We also identified the terms genotype, phenotype, and we also introduced the terms homozygous. We already introduced the term homozygous and heterozygous. We talked about dominant and recessive. Okay, so we covered through the first, looks like 6 or 7 bullets of our objectives for this semester, for this chapter. Where we left off was getting ready to prepare a Punnett Square for a monohybrid cross, and then I've kind of pushed you a little bit over the weekend to look at a dihybrid cross. And we're going to go through and do each of those today. And then we're going to step back and then talk a little bit about Mendel. We skipped over him because I wanted to give you the terminology to work through your handout. Okay? And then I'm not sure if we'll fully get to it, but we'll identify that these variations that we see in generation is due to the mix-up, if you will, of chromosomes during the processes of meiosis. So we're certainly putting into practice that Unit 3 material right here for inheritance patterns. Okay. And then probably on Thursday, we'll talk about pleiotropy and phenotype. So I'll give you guys some additional handouts for the remainder of Chapter 10, but today we'll just look at how do we make a Punnett Square if we're looking at 1 or 2 traits. Okay, so on Thursday, we'll probably get to these things. And then also to Chapter 11, which is getting into evolution, I think is, chapters might be off a little bit. But hopefully we'll get into some of Chapter 11 maybe on this upcoming Thursday. But let's go back to our handout. And we were talking about, we mentioned, we didn't really talk about him, Gregor Mendel. What did you guys find out about this guy? Anybody heard of him before? He's a monk. He's from where? He did discover the principles of heredity. And so we're going to see his important work gave us this foundation of inheritance. I think, to me, it's interesting to point out in a class like this. What's a monk? What is a monk? A religious guy is taking a vow. Okay, so a religious guy studying what? Science. Okay. In America, we hate the concept that science and religion could have anything at all similar at all. But really, if you go back in time or any other country, right, there's religion and science really do go thoroughly hand in hand. So I think it's neat that there's a religious guy doing some really cool genetic studies back in the 1800s. So he is from Austria. He is a religious monk, and he did, as identified, study inheritance, patterns of inheritance in the mid-1800s, so a few years ago, a couple of years ago. It's important to understand, even though he did give us these important foundations about inheritance patterns, he didn't know what you guys know. You may moan and groan all through biology, blah, 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 chromosomes, DNA, 5', 3', whatever. He had no idea about those things. He had no idea what DNA was, its structure, its importance, etc. Yet he's studying these inheritance patterns that are strictly dependent upon DNA sequences. He had no idea that DNA could be arranged in chromosomes or sorted during meiosis. Right? No idea about the role of chromosomes in DNA, as well as the role of meiosis. But he was setting the stage for our better comprehension now of the role of DNA arranged ancient chromosomes that can be separated during a process called meiosis. So he didn't just making observations of the phenotypes of different species. Does anybody know what he was studying? The pea plants. Okay. And here's an image I got from the DNA Learning Center, the online repository of his garden that he actually did his little study. So it's not real laboratory setting. It's just a nice little pretty outdoor garden that he was making some nice scientific observations between one plant generation to another. I was reviewing this, and I didn't give you guys much room in here, so we're talking about his contributions that are right small or maybe on another piece of paper. I apologize, I'm trying to guesstimate, but looking at it in better detail, there really isn't much room. But there's 4 primary things that we can kind of take away about what Mendel did for us for understanding inheritance. One is, he said, the basic units of inheritance has to be something physical. He said there is something physical about these plants that I'm observing that is being transferred from one generation to the other. Right? Or as identified here, material element, something physical is being passed from parental to offspring. And leave a little space after each of these. I'm going to add a term that we now know is a proper term. And so we're going to say, well, this is what he said. We're going to give a term that describes this, and this physical something. So in addition to identifying that inheritance is due to something physical being transferred from one generation to the other, he said these physical elements, they must come in pairs. They must be found in pairs. So there's something physical that's transferred from generation to generation, and those physical elements come in, they must be expressed in pairs. Thirdly, some characters may be retained throughout generations, right? So not just from parent to offspring, but to what in a human we would call grandchild or great-grandchild, something like that. It's not just this one generation transfer, it's retained throughout the inheritance of that particular lineage or line of breeding patterns. So this material element comes in pairs and it's retained over generations. And fourth, he said during the formation of the plant pollen and the plant eggs, these pairs got separated. Bless you. Okay, so they're not only coming in pairs, but when the eggs and the pollen are made by plants, those pairs are actually separated into different eggs or pollen granules. Okay? Those pairs are there, they're retained through generations, and they can separate in order to make a seed in the plant that he was studying. Okay, to move on. And really what he did is he gave us the basics, which we identified already, for understanding how physical characteristics are inherited right, from one generation to another, as I've been stating.
You said these pairs separated when?
During the formation of, the word I have there is gamete, but during the formation of, what he was looking at, the plant egg and the plant pollen granule, the gamete. You could say gamete, or you could say pollen and egg. You want to be specific to the plant. Is that okay? Got it? And so he's just giving us, as we identified, the foundations for inheritance of characteristics from one generation to another. It's kind of a founding father, if you will, for inheritance. And we already identified that he studied pea plants. Okay, so here's I don't know where this image is, but it's on an old anatomical drawing of the pea plants, looking at the fertile flowers and the rest of the growing plants. And here's kind of a more modern image of a pea plant, so the garden pea. There's just like there's many different types of dogs or cows or horses or bears or whatever. There's many different types of peas that offer genetic variability. So he was studying a garden pea, which genetically is not too complex compared to something, say, as yourself. Yes? I can. Right, so he was working with the garden pea, and if you read ahead, why did he use, what about the pea plants? What's so special? They're easy to grow. They produce many offspring or many seeds. They develop quickly. So they're easy to grow. They produce many offspring. Their traits are not very diverse. So you guys identified. Yep, they're easy to grow. They're short lifespans. Yes, they grow quickly. Yes? Very good. That's important to add as well. I don't know if I have it up here, but they're very easy to control their reproduction by moving part of the reproductive structures of the flower. Okay. Their reproduction is easy to control as well. Their characteristics, their traits that he looked at were pretty simple. It was like either one or the other. Okay, so nice little subject to study. I do kind of have that here. Easy to mate or crossbreed, cross-pollinate. I've identified. A nice little subject for someone who's a garden to go out and study and make some observations about inheritance patterns. So the pea plants. If we look at the basic structure, y'all studied flowering plant structure, didn't you? So you know where the ovary, what is found in the ovary? Ovules, little eggs, okay, that have gone through meiosis. In the anthers, there are what? The male reproductive, the organs, and even the sperm, or we call it pollen, okay, so that have also gone through meiosis to be produced. So a little bit of genetic diversity. If we look at some plants can be considered to be true breeding plants, and these plants have the capacity, will learn to self-pollinate. And others are considered to be hybrids, and I'll come back and address those. But first, if we look at true breeding plants, these are the types of plants that can self-pollinate. What does that mean? They can fertilize themselves. They have both male and female structures. They have both pollen and ovule, egg. In our true breeding plants, the traits that you'll see should be homozygous. Whether it's homozygous dominant or recessive, you'll continue to see the homozygous characteristics exhibited. There's not much variation here. The self-pollinating plants. If we contrast the true breeding plants to what we can call hybrid plants, or those plants that are a result of cross-fertilization. What does that mean, cross-fertilization? Two different species. They've got to be manipulated, or there's got to be a carrier. Okay, there's some bee or a human or something moving the gametes from one plant to another, and then kind of crossing their genetic material. Okay, so if we look at a plant, it's a pollen probably being transferred from one plant to another. If we look at hybrids and cross-fertilization, that's when we can begin to see some heterozygous traits expressed. For a simple answer, yes. That's going to go into more detailed discussion like we kind of dabbled in last Thursday, where we looked at like the variation of like our human skin pigment. We're not just black or white, we're shades. And so not all genes we're going to learn are as simple as A or B. And so that blending or variegation is a blending kind of pattern. But you're right. It's more into the if you remember, how did we define a heterozygous? Yes. In a heterozygous trait, we know that the dominant allele is expressed, but there's certainly going to be a recessive allele retained, and so the offspring have the ability to express which strain eventually. Recessive. Either one, but eventually there's a chance for the recessive trait to appear in some of the offspring. Not all of them, but some of them. If you look at this terminology and take it away from a plant and look at, for example, like we're in the cattle business, they call it like hybrid vigor, or maybe they use the same terminology in the horses or dogs or any kind of animal that you may show or raise. When you get into this heterozygous trait, sometimes the animals or the offspring are a little bit more genetically diverse, a little bit genetically advantaged for whatever character you're trying to select for. So, for example, in the cattle business, some people will breed animals that are originally from India, and India, if you know, is warm. Okay, so those animals are adapted to warm environments, and we can cross those with animals that are from European continents that are a little bit more temperature-wise cold, but they grow and get more muscle. So we can cross a European versus an Indian breed of cattle, and these cattle will be more musculature but able to adapt to our heat, okay? Because they're dry environments, because they're hybrids, is what they're considered. So we can look at it not just at plants, is where I'm going with that scenario. Okay, so true breeding plants are self-pollinating and continue to see that homozygous trait consistently expressed. So if we begin to cross-pollinate or cross-fertilize, you can begin to see some of the recessive or homozygous vigor and expression, some variation in the offspring. And so what Mendel did is, in figure 10.2, illustrates how complex the scientific studies were. Not very complex. He took scissors, I think, or a knife. He cut off reproductive structures of one plant and then pollinated another plant to create hybrids or cross-pollinated the plants, and then planted little embryos. We wouldn't call these embryos. What would we call these? We call them seeds in a pod. But they're embryos that could grow into a mature plant themselves that would go on and reproduce. All right, so I'm going to go back to that list that I didn't give you much room to add in there. And we identified, we said, based on Mendel's work, he said the basic unit of inheritance are material elements. Okay, so something physical about plants is being passed from one generation to another. Okay, the physical something that we now know is called genes. Genes are transferred from one generation to another. They're inherited. He also identified that these genes may come in pairs, and these have now, his terminology has been now called those homologous chromosomes that we saw lining up during meiosis. They're replicated. We now call those homologous chromosomes. Yes, they do come in pairs. And then tying right into it, he said these characters are retained through generations. when we know that our chromosomes are retained, right, from one generation. Our chromosome numbers are retained, right, consistently, right, from one generation to another. And where would we see these homologous chromosomes separating? What is the name of the process to better clarify that fourth point? Meiosis. Okay? Meiosis. So Mendel set us up for pretty accurate foundation of inheritance patterns, giving us now the ability to define a gene versus a chromosome versus a homologous chromosome, and then how they separate during a process called meiosis. If we go back and look at the pea plant that he was studying, it really was kind of A or B for the traits which he was studying. He was looking at some characters of the plants, looking at the seeds and this pod and the leaves and the height of the plant and the color of the flowers. And if you look at some variation, you look at the seeds, if they were in the pod were smooth or wrinkled, or if they were colored yellow or green. What else? If the pod shape was more, is described here, inflated, or more kind of shrunken. Other traits, the pod color could also vary, green or yellow. The flower was produced was either purple or white. And the position of the flower was at the end, right, or on the stem, some variation they saw. And then he called some plants tall or dwarfs. I guess that really is like textbook 1A or B. What we're going to learn is not everything is as simple, which is good, that we're not as simple as a pea plant. But some other species aren't as simple. But it was a great species for him to study and let us understand the basics of inheritance. I want to show a quick video of someone who is doing some plant cross-pollination. So just to put it into visual perspective.
Early in its history, the
Is that
Earth
okay?
consisted of little more than land masses and the oceans in between. But all that changed when simple cells, the first living things, formed in ancient waters. Some of these early life forms could make their own food using sunlight. Over time, their descendants evolved into plants. Plants capture light energy from the sun and convert it to chemical energy through photosynthesis. This is possible because of chlorophyll, a chemical in their leaves and stems that captures the sun's energy. As botanist Warren Wagner explains, since the rest of us can't use the sun's energy directly, we eat plants to get the energy we need.
So basically, they're the food for all the world. And I consider biology the study of plants and their parasites. And everything else is a parasite on plants to eat. We do. If we don't eat the plants, we eat something that ate the plants.
We depend on plants and their ability to reproduce.
Plants require two special things. One is to reproduce themselves, to occupy space. And the other one is to change. You've got to change because there's changes in climate, changes in rainfall, changes because of volcanoes, changes because of new kinds of organisms. And in order to be in this melange of all these constantly changing things, you're going to have to change yourself.
Plants reproduce and change, but that's complicated because they remain in one place. Most are bound to the ground by roots. To spread from one place to another, they often send out spores and seeds.
I picked the wrong video. This is not looking, that's obviously relevant, but I wanted to show you the cross-pollination videos. Let me see if I can find that for you real quick. This is what I wanted. Sorry about that.
Cultivation of naturally colored cottons has the potential to reduce the need for pesticides and chemical toxins. cultures used colored cotton for textiles for centuries, but commercial production of the plant started in the U.S. only recently. Because of low yields, short fibers, and the availability of cheap dyed alternatives, naturally colored cotton didn't get much attention here until Sally Fox began addressing the problem in the 1980s. Fox was an insect biologist when she was confronted with the challenge by a local cotton breeder to develop commercially viable colored cotton plants.
I just looked at this and said, well, what's going on here? Why isn't anyone improving this fiber so it could be more easily spun? And he said, well, there's no market for it. And I said, well, why isn't anyone making a market for it? And he handed me this stack of seeds and said, well, why don't you do it?
So she did. The first challenge was to breed cotton plants with longer fibers that could be machine spun for a commercial market. Sally worked on developing such plants by cross-pollinating various plants selected for their fibers and colors.
This is the female part of the flower right there. That's the stigma. And we've taken the pollen off of this flower last night. Now I'm bringing pollen from a different plant. You can see it's a completely different flower. And putting that pollen right on the stigma. Now, the seeds that develop in this cotton boll, this one boll right here, will be hybrid seeds and they're the beginning of a lot of potential new variability.
The new colored cotton plants have the same advantages that native hand weavers have appreciated for centuries. There is no waste produced by dyeing, and the plants are naturally resistant to pests, so pesticides aren't needed.
The cotton plants that produce this cotton of color are more vigorous than regular cotton. They're from wilder origins. They haven't been domesticated and put through breeding programs where people are using pesticides.
What's more, the greens and browns produced naturally actually intensify with washing. Still a fledgling industry, other breeders are attempting to develop more colors, but the market for this cotton is still limited. But hopes for the environmentally friendly product are still alive. Naturally colored cottons are used in the manufacture of cloth for the natural products industry both here in the U.S. and in Europe.
All right, I just wanted to show you someone doing hand pollination or cross pollination. So putting it into a wheel, not just something that I speak. Just to recap for you, some terminology that we covered on Thursday. We talked about an allele, and if you remember, we said that's just basically a what? Different variations of a gene. Okay. We also identified the difference between a dominant versus a recessive. We learned that dominant traits or genes or alleles or what? Wherever they are, they're shown, they're expressed in the what? How could we describe that? What it was? Yes, you're right. And what term did we use for a trait expressed in an organism? The phenotype. Okay. So if there's a dominant allele present, we'll see it expressed in that organism's phenotype. When will we see a recessive phenotype expressed? Very good. When only the allele variants that are inherited from the parents are recessive, or we could say homozygous recessive for that particular trait. Letter-wise, how would we represent a dominant allele? We'll use a capital letter, and then a recessive we'll use a lowercase letter. And I don't think I put it in your handout, so right under dominant allele, recessive allele, there's a term that your book identifies, which is important, which is called a wild type, and it illustrates the concept that we discussed on Thursday. If you remember, we said just because a trait is dominant doesn't dictate its expression in the environment. The scenario we gave about the people who live in a Scandinavian country, although blonde hair, blue eyes are recessive traits, those are more expressed. And so the proper term for illustrating a trait that's the preferred trait expressed in a population is called a wild type allele. A wild type. You can talk about a wild type allele, wild type genotype, or wild type phenotype. But it's the naturally expressed favored allele, genotype, phenotype in a population. Just whatever is favored for that population. Does that make a little sense? The wild type is just what's naturally expressed in the environment for a particular population. We also identified, we gave some examples, just a few examples of human dominant traits, but in some further examples, freckles are more dominant, but do we see a higher proportion of freckles in our environment? No. So even though it's a dominant, it's not the wild type in some populations. The widow's peak, the crown of the head. Your earlobes, whether they're free or attached. Okay, so the free earlobes would be the dominant. So you can do a very simple genetics study by looking at your parents and your earlobes, or look at your mother and your alleged father, and and then your earlobes to see if it makes genetic sense. The little finger, if it's bent, is actually you don't have a problem. It's a dominant trait as well. Short big toe is a dominant trait. The ability to roll one's tongue is an example of a dominant trait. The hitchhiker thumb, where it flips back, dominant trait. Cleft chin, dominant, but again, not necessarily something you see real common. It's not the wild type expression. The finger hair, dominant trait as well. Okay, so there's some examples where we can look at dominant and recessive traits in humans. Also, on Thursday, we identified genotype. So I'm just going to quickly go through this. We already identified it. We said that's just the allele combination, whether it's homozygous, dominant, homozygous, recessive, or heterozygous. Just the combination. And we identified if a homozygous dominant, when we filled in our chart, we said we just have the allele variants would both be dominant. If we look at homozygous recessive, both alleles would be recessive. And the heterozygous organisms, we would have a mixture in the genotype. The genotype then dictates the phenotype, which we also defined on Thursday, so I'm going to move through a little bit quickly, and that's just the observable physical traits dictated by the genotype. So the genotype determines the phenotype. And that's about where we left off with some of our terminology on Thursday. You guys are still writing, so am I okay to move on? Or just want to review those. Where we did not discuss are the different types of generations. And if you're trying to do a genetic study, and if you're going to do a simple one at your family, we can define, you've got to have a starting point somewhere. Whether it be at your parents or your grandparents or you, whatever, there's got to be a starting point, so we have a point of reference. And so the first generation that you're studying is called the parental generation. And you'll abbreviate it with the letter P. And so the parental generation is the first generation that's being studied. You can look at it, Mendel used it for his plants. You could look at it for your dogs, your trying to study, your kind of starting point would be the parental generation. When the parental generation mates or crosses or breeds, whatever term you want to use, they produce their offspring. And their offspring collectively are called the first filial generation. And you write that F subscript 1. Okay, so first filial is the offspring. So if your grandparents are the parental generation, your parents would be the first filial, and then you would be the second filial, okay, or the F2 generation. The F2 generation is the offspring of the F1 generation, and it just goes in sequence. And now we're going to use these terms depending on our practice, so looking at those monohybrid and then dihybrid crosses. So we're going to first do the monohybrid, and then we'll look at the dihybrid. That's when you can look at your handout you're going to submit today. So if we look at creating what's called a Punnett Square, it's important to realize it's just a tool. It's not a guarantee. It's just a predictor to make some analysis. It's just a very simple genetic tool to understand how a particular trait or multiple traits are passed from, say, the parental to the first and then to the second filial generation, or how they may not be. So it's just a resource to help predict, but there's no guarantee. I did my Punnett Square, it says all my kids are going to be, there's no guarantee, but it's just a nice little resource. The first type of cross that we're going to do is called a monohybrid. So mono means one. We're just going to look at one trait is what we're looking at. We're going to cross two parents, breed, two parents, parent plants, for one particular trait, a monohybrid cross. And then we'll contrast that to a dihybrid cross where we're looking at two. I could go exponentially here. I could look at the relationship of 3, 4, 5, 6, 7 traits, but you notice how difficult when it was with two traits, that if you start adding multiple traits, there's a lot of complexity in interpreting the results and making sure you're doing it correctly. And so the first thing is, it's important that you create a legend that's legible and sensible. So your handwriting is important in this case. And I also gave you a heads up on Thursday. I wanted you guys to bring some different colored pencils. Maybe you want to get those out, or if different colored pen. If you have them, if not, just write really clearly in your notes so you're not getting confused. So we're going to create a legend. And so we're going to do a scenario of a gene called lactase. Does anybody know what that's lactase? Has something to do with that? Let's work backwards first. So ASE is an enzyme that has something to do with milk, the sugar, and lactose. Okay? So lactase must do what with it? Digest it. Okay? It actually splits it. Lactase is an enzyme that actually splits milk sugars so we can digest it, right? Absorb it and then metabolize it. If it's present, we can use lactose. If it's not present, what happens? You're lactose intolerant and you're sick. Okay, when you eat any of the sugar, because you can't break it down. Okay, so we're going to look at this expression of lactase. What letter do you think would be useful in this scenario if I'm trying to look at this? Letter L, lactase. Okay, so if I want to study the dominant allele, I would use which letter? Uppercase L. Okay. And the phenotype associated with this dominant trait is what? having the enzyme, and then hopefully lactose tolerance. Okay, so if that allele is present, we should expect that enzyme to be expressed. Recessive allele, I would indicate. Very good. Lowercase l. Don't use a completely different letter for I for intolerance or whatever. It needs to be the same letter so you're not getting super confused. Okay, so just do a lowercase l, and of course in this typing it does look like an I. That's a lowercase l. And if the recessive allele is present and it's actually phenotypically expressed, we would say this person is lactose intolerant because that enzyme is not expressed. Okay, so no lactase, and then person will know if they're lactose intolerant. They don't have to do a test. They know. Eat dairy, get sick. For a nice week. That's okay. All right. So we're going to go through and do a monohybrid cross. We're just going to make up a scenario. In a monohybrid cross, we first created our legend, and then we need to do a cross. And it's important to understand if I'm looking at the parental generation, if they're homozygous, dominant, homozygous, recessive, or heterozygous. Let's just make one up where we know the genotype of the parents. Okay, so we're going to predict the allele combinations at fertilization. So we're going to create a baby, or potential babies, based on the sperm combinations of the father and the egg combinations of the mother. Right, where we have 4 possibilities of gamete formation. Okay. Well, let's just create a male. Now, before I get too carried away, we're looking at allele combinations at fertilization. So the male brings what to the party? Sperm or pollen. In this case, we're looking at humans, so sperm. What type of ploidy? Haploid. So notice that when I'm looking at the fertilization, I'm just going to look at haploid. I'm just using one letter. If I see one letter represented, that tells me that's a haploid cell. So part of the male contribution. In this case, genotypically, if we look at the parent producing this person would be a heterozygous person. That's both dominant allele being passed and recessive allele being passed. But I'm not mixing them together because the sperm are haploid. We're going to do the same thing with the female. We're going to use a female that's also heterozygous. Okay, so half of her eggs will have which allele? Or I could say dominant, and the other half would be recessive. And I'm going to use some different coloration here so we can see. So half of a heterozygous woman's egg pass on the dominant allele, half of them pass on the recessive allele, and the males the exact same thing. So we're crossing 2 heterozygous parents, both that express the lactase gene. But we're trying to see, well, would their babies all express the lactase gene? And in order to create a Punnett square, it's just like a multiplication table. And so we're trying to figure out the combination of a diploid organism, or the offspring. So how many alleles should I have expressed per potential offspring? 2 alleles, because the offspring would be diploid. They'll get half of their alleles from mom and half from dad. And so you'll just bring down the alleles for father, and then you'll bring across the alleles for mother. We'll look at some potential possibilities. So in that case, in the first scenario, the dominant allele for the father, I'm going to keep in purple, and the dominant allele for the mother. Okay? So 25% of their offspring are going to be considered to be homozygous dominant. And you just keep going through. Okay? So go ahead and practice with the scenario. Finish this little table. If I continue on to look at the other combinations, still bringing half of the dominant alleles for the male, and then crossing those then with the recessive with the female, and then doing the same thing with the recessive allele for the male, and then continuing on. We can go through and see that the genotype of all the offspring is not going to be identical. And we have a nice combination because at fertilization we get genetic diversity. And we begin to see this with just looking at one gene of the thousands that our bodies have. So now we have an idea. Before I move on, can you see how we fill in the table? We try to do a simple mating to look at the offspring. Can you see the difference between the diploid cells, the result of fertilization, versus the haploid cells that are used for fertilization? And now you can interpret the genotype. So go through in your box. You should have room, okay, or in the next table, excuse me. Interpret those genotypes, right? Interpret those in the form of a genotype. So this corresponds, first off, to this keeping the relationship with that. Just write in there the phenotype. You would say homozygous dominant causes which phenotype you would see, gene present or not. So in the first box, or the first offspring, where the dominant allele from each parent, what would the phenotype be? Homozygous dominant, and the gene is lactase is present, it's expressed. What if we go to the next scenario where the male contributes a dominant and the female contributes a recessive? Heterozygous, and we still see, moving the wrong way. Okay, so in this scenario, we'll still see the lactase present. Let me go over to the third column. I've got that showing next. Okay. In the next scenario, the female is contributing a dominant allele, and the male is contributing a recessive. So we would call this a heterozygous, just heterozygous, and the trait is or is not expressed. It is expressed. Okay. The other scenario, the far left corner, we just covered. And in the last box, we have two recessive alleles, so we would call this homozygous recessive, and lactase is not present. And so if that child is born, they would be considered to be lactose intolerant. That's a simple, if you will, monohybrid cross. The important thing is to create a good legend so you can go back and use it for your interpretation. As well, understanding when you're looking at a gamete versus producing a potential baby offspring, haploid versus diploid. What questions do you have about that before we move on and step up the game? All of it, right? Looking at a dihybrid cross. Let me go look at your interpretation. There you have the where we just finished. And if you look at the percentage-wise, of the phenotype, what percent of that table express the gene? So just going back. The dominant phenotype is expressed of the 4 potential offspring how many times or how much? 3 or 4, which is 75%. What percent is recessive? 25%. So it's based on Mendel's study, he consistently saw that 3-to-1 ratio. When he, every now and then, it's about 25% of the time you can see that recessive trait. That's kind of like a cogs turning for all these things. Like, where is this coming from? It's through the meiosis combining our recessive traits and allowing them to be expressed during fertilization. So if we're looking at crossing 2 heterozygous organisms, you'll consistently see probability-wise this 3-to-1 ratio of the homozygous, the dominant trait to the recessive trait. If we look at what that's contributed by, it's due to what's now called Mendel's law of segregation. And we get some variation, right, of different traits where this 3-to-1 type of probability because of our what we call now alleles that separate during meiosis. Okay? So that we know that our gametes only get one of those alleles. This is restating what we used to set up the table. But Mendel was concluding this. And now we're putting it into practice. There's different traits of organisms, those different materials that are inherited, they would now call alleles. They're actually separated during what we now call meiosis. That's how we set up our haploid cells across those during our little Punnett Square to get the offspring. In other words, we're getting variation in offspring because of meiosis, mixing up those alleles and allowing for every now and then that 25% of recessive trait to be expressed in the population. And it's just called Mendel's law of segregation, tying to how our alleles do separate during meiosis 1. During the smallest chromosome separated. The follow-up question that is asked is, are our traits really, let me come back to those, are all our traits really that simple? No, they're not. Okay, all of our traits, some are, but really not. Most of them are not. Okay, most traits are really not this simple. And it just tells us how some traits are controlled by one gene that has 2 separate alleles. Repeating what we started introducing on Thursday. It just tells us how one gene just has 2 variations, but really most genes aren't that simple. We'll look at a scenario in just a moment where we're going to do a dihybrid cross, where we're going to look at 2 traits and then kind of step up the game as far as genetic diversity. Are we okay to look at, move on? Get a dihybrid cross. Oh, sorry. The term for these simple traits that we've given are called Mendelian traits. So the really simple ones where there's 1 gene, 2 variations, there's nice and cut and dry, those are called Mendelian traits. There is a website that you might want to reference here as well. It's called the Online online Mendelian inheritance in man. OMIM, I think is the name of it. Online Mendelian inheritance in man is a really cool website where you can look at some what's considered Mendelian traits in humans. It's pretty. It's got a ton of information. It's got sequences and variations, all kinds of information. But if you get some time, if you're curious about this, there's some a really cool published website of different human genes that have this Mendelian trait where there's one gene, two variations, on and off kind of expression. We're going to then create a dihybrid cross, and then I'll walk through it, and if we have time, we'll look at a little animation to put it into perspective as well. Hopefully put it into perspective for you. So if you're looking at a dihybrid cross, it's going to get a lot more complex because we're not looking at just one, we're looking at two traits. And so we're going to fill in your little, the rest of your handout, kind of like what you did with handout that you're going to turn in today. We're going to first set up a legend. If you're able to print page 5 and 6 of your handouts, I've got a list of important tips to keep in mind when you're doing a dihybrid cross. So you don't have these today. I'll read through them without writing them down. I've got them listed for you on page 5 of your handout. One important thing is, like I mentioned earlier, to continue to create a legend. And make sure it's clear and legible. But we're creating two legends, right? We're looking at two traits. So we still need to identify the dominant trait, dominant alleles, dominant phenotypes. Not just once, but twice. You also need to remember, as we identified earlier, to use the same letter. This is really important, even more so when you're looking at a dihybrid cross. So if I'm looking at lactate phenol, I'm going to use L. If I'm looking at hair color, I would use H, right, for dominant and recessive. So make sure you're not having 4 different letters for 2 variations. An important step as well is to also create the possible alleles in the parent gametes first. So you're going to, before you can even do the cross, you got to create the parent gametes, which would have what type of ploiding? Haploid for each allele. So there's going to be 2 letters, but the letters will be what? Different. They'd be different, okay? But there would be 1 letter of each. 1 A and 1 B. It wouldn't be A and A, or B and B. It would be allele A, allele, different gene, B. So even though we're looking at 2 alleles, they're different. So in that case, those alleles are haploid. So we got to create our haploid gametes first, for both the mother and the father. So we have to have an idea about whether they're homozygous, heterozygous for whichever trait. Once we create the haploid sex cells, the sperm and the egg, then we can put those into a table and cross those to create what type of offspring? Ploidy-like. They would be diploid. And so in that case, we would have 2 alleles for each gene. So we actually have how many letters in our columns for our offspring in a dihybrid cross? There's 4. 2 for one gene and 2 for the other. Good. Again, these little tips that I have are on page 5 of your handout. We're going to do a scenario. We're going to look at 2 genes in the human. Okay, so the first trait we're going to look at is tongue rolling. So on your handout, if you've got it, trait number 1 is going to be your tongue rolling ability. We'll just use the letter R for the ability to roll your tongue. And that'll be a dominant trait. We'll use the lowercase r for the inability to roll your tongue. You can practice this at home later if you do that or not. The musculature in some people's mouths is such that they can do that or they cannot. Okay. So that's going to be trait number 1, the ability to roll your tongue or not. Dominant ability to roll, recessive inability. The second trait that we're going to look at is the presence of freckles. Okay? And so dominant would be the expression of freckles, and recessive would be no freckles.